Okay, ladies and gentlemen, you are welcome. So in this video, we are going to look at the solution to chemistry of solution to past question paper on chemistry of macromolecules. I hope you can all hear me. So let's look at okay, please, can you hear me now? Hello, can you hear me now? Hello, can you hear me, please? Okay, good evening, Messi. Can you hear me? Okay. So please, what I want you to do now, go to the, I want you to go back to the group and tell them this is the one that we change another link. So this is the link that we are going to use. So please, can you hear me? Hello, can you still hear me, please? Can you hear me now? Okay. So please go and tell them this is the link that you are going to use. So without wasting much of my time, let me share the question paper that we are going to use, sorry, that we are going to solve. So I'm now going to share the question paper that we are going to answer. So please, I don't know if you can clearly see. Please, can you clearly see this question paper? Hello, can you see question paper? I just shared my screen. Okay, so what is the problem? Okay, not clear. So let me share it again. So now this is the question paper. And I think what I'm going to do, I will be reading the question so that at least, even if you are not seeing it very clear, I think I will be reading the question. So this is biochemistry question paper, and it is 2023 January examination. And these exams, there are 6 questions. There are 6 questions in total, and you are to answer 4 out of the 6 questions. So you are to answer 4 out of 6, and the timing for the exams is 2 hours 30 minutes. So it's 2 hours 30 minutes. So I'm going to actually try as much as possible to answer all these questions here. So I'm going to answer the question together with you. So the question 1A says, draw an animal cell showing 5 organelles. Draw the animal cell showing 5 organelles. So it means that, ladies and gentlemen, you should understand that it is very important as a student that is offering biochemistry to know how to draw the structure of animal cell. Remember, let me briefly talk about cell. What is a cell? A cell is actually a structural, it is a structural and functional unit of life. It is a structural and functional unit of life. So when we said a cell is a structural and the functional unit of life, it means that anything on this earth that is considered as a living thing, it means that it must possess a cell. So without a cell, there is no life, and that is why we have a different uh, cellular theory. One of the cellular theories said that there is no life apart from the life of cell, meaning that if there is no cell, there is no life. So therefore, all living organisms possess cells, and if there is a problem with any cells, it means that there is a likelihood that there will be a problem with tissue. Because remember, from the concept of cellular organization or organization of life, life started from cell into tissue, then tissues into organs, then from organs to system. And then the system, from the system, you have a complete living organism, a complete living organisms. But remember, ladies and gentlemen, life doesn't start only from a cell. It started from chemical level. Chemical level. And remember, what are these chemical levels? I think in one of my videos, I told you that life started from atom. Atom form a molecules. Molecules form a complex molecules like carbohydrate, proteins, and those proteins, carbohydrate, form supramolecular complexes and the organelles of a cell. And remember, we have types of cell. Please, who can tell me what are the types of cell? 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 Okay, excellent. We have prokaryotic and eukaryotic. So actually, ladies and gentlemen, this class we are going to have like a general revision of the course. So what is prokaryotic cells? It means that they are the cells that doesn't have nucleus. They don't have, that is no nucleus and membrane bound organelles. You see, is it marvelous? Please, at this stage, marvelous Idara Friday, I was shocked and surprised that still at this stage, we are expecting that the types of cells is plant and animal cell. No, the types of cell is prokaryotic and eukaryotic. Please, you shouldn't forget about this. And whenever we say prokaryotic cell, prokaryotic, it means that it is a cell or are cells that doesn't have nucleus. They don't have nucleus and membrane-bound organelles. What are nucleus? Sorry, what are membrane-bound organelles? Mitochondria, Golgi body are examples of membrane-bound organelles. So when we say prokaryotic cells are cells that doesn't have nucleus and membrane-bound organelles. While in the case of eukaryotic, they have, in this case, they have nucleus. They have nucleus and membrane bound organelles. So please, you shouldn't forget about this. So whenever we say types of cell, we have prokaryotic cell and we have eukaryotic cell. Prokaryotic cell, no nucleus and membrane organelles. Then eukaryotic cells, Yes, nucleus and membrane-bound organisms mean that they have nucleus and membrane-bound organisms. So who can give me examples of prokaryotic cell? Examples of prokaryotic cell. Examples of prokaryotic cell. I want to be having like a kind of discussion so that at least at the end you will be able to enjoy uh, the solution to the past question paper. Yes, yes, yes. Excellent, excellent, excellent. Examples of prokaryotic cell is bacteria. And another examples of prokaryotic cells, fungi is not a prokaryotic cell. Algae is not a prokaryotic cell. Rather, bacteria, we have archaea, archaea bacteria, archaea bacteria. So these are the two. Bacteria and archaea are examples of prokaryotic. Please, at this stage, Vivian Johanna, virus is not a complete living organism. Rather, it is called a particle. Please, you see, this is how many students are failing exam. Fungi is not prokaryotic. Then examples of eukaryotic organisms, that is those that have nucleus and membrane-bound organelles. We have, who can give me examples of eukaryotic cells? Eukaryotic cells, please. Examples of eukaryotic cells. You see, it is from this video that you will be able to pass this course. So that is why you should be very careful. If we can be able to answer all these questions, so I think we will not have a problem. So eukaryotic cells, even human cells, like plants, plant cells is an examples of prokaryotic cell. The fungi, the fungi, algae, they are all examples of prokaryotic cells. And then the next thing is classification of cell. Classification of cells. So please, who can tell me what are the classification of cells? We also have two classification of cells. Please, who can tell me what are those two classification of cell? Two classification of cell. Archaea bacteria, archaea, archaea. That is another examples of prokaryotic. So now I am asking about classification of cells. Please, who can tell me what are the classification of cell? Excellent, Grace Momo. We have unicellular, and we have multicellular. So you see, from the word unicellular, it means it means they have one cell. And multicellular, it means they have many cells. So unicellular, they have only one cell, and multicellular, they have many cells. So these are the two classification of cells. And what are the examples of cell? Who can give me examples of cells? Examples of cells. So examples of cells. Who can give me what are the examples of cells? Examples of cells. What are the examples of cells? Examples of cell. Excellent. Plant cell, animal cells, no, plant cells, farm cells, all are examples of cell. Muscle cells, brain cells, excellent. So now we are what some of the examples of cells we have, as we can see, we have plant cells and animal cell. We have plant cell and animal cell. So I think if you can read from the question, animal cells, that is the cells that is from animals. So the question says, what are the organelles that are found in animal cell? In animal cell. So at this point, you must know how to draw the structure of animal cell. So knowing how to draw the structure of animal cell and plant cell is very, very important. So how do we draw the structure of animal cell? So first, the first thing that will come into your mind is that animal cell, it is spherical in shape. It is spherical in shape like this. So it should be spherical. So that is the first thing that need to come into your mind. So the first thing is that animal cells, it is spherical in shape. And the question says we are, let me share the question again so that you can see the question clearly. Let me share the question again. Look at the question very well, please. You see, the question says draw an animal cells showing any 5 organelles. Draw the structure of, look at the question very clear. Draw animal cells showing any 5 organelles. So now, so this is first you need to draw the spherical shape or the cycle like this. So 5 organelles. 5 organelles. So how do you draw them? First, we have nucleus. So we have the nucleus here. I hope you are seeing how I'm drawing. You see, first, we have nucleus. Then second, I was able to draw nucleus. Then I have something like this. Please, you can tell me what is this in a cell? Something like this. What is this, please? Who can tell me? Okay, what is this? It's not DNA. Excellent. It's mitochondria. Mitochondria. So you see now I have 2 organelles already. We have nucleus and mitochondria. Then who can tell me what is this? What is this place? Excellent. We have now endoplasmic reticulum. Endoplasmic reticulum. Reticulum. Okay, then what is this? So the first thing you need to know how to draw the cycle. So now, who can tell me what is this now in a cell? This is not ribosome, it is a lysosome. Lysosome is lysosome. Okay, so now we have 1, 2, 3, 4. Then okay, now the ribosome is here. This thing that you are seeing here attached here, this is ribosome. Ribosome is usually attached to endoplasmic reticulum. So this is the ribosome. This is ribosome. So you see here, we drew the animal cells showing the 5 different organelles. So it means that now we answer question 1A. Please, I don't want to be wasting much time. Someone is saying to my board is not clear, but some of you are seeing the board. Maybe the network is from your side, please. So now we are able to answer this. So let's look at question 1B. State the functions of 4 of the organelles in A above. The functions of the 4 organelles in 5 above. So now, oh, excellent, farm. So now we mentioned 5. So we want to know the functions of 4 out of them. 5 of them. So you see, this is how to answer exam. So see, if I am the one that asks like this question, draw the structure of animal cells and show the 5 organelles of the cells, the first thing that I'm going to do is that, okay, that is how to pass examination, but that is how to pass examination. First, you ask question 1A, how to, the question says indicates or draw the structure of animal cells and show 5 organelles. So if I'm the one to answer this question, the first thing that I'm going to do, I will start with defining cell. Cell is the structural and the functional unit of life, and we have different examples of cell, like plant cell and animal cell. So then you answer E. Animal cell is spherical in shape. Then you draw the spherical shape of the animal cell, and then you indicate the 5 organelles. And then you move to B. Move to B. So the B, we are to actually mention the functions of the 5 organelles. So now we have 5 organelles here. What is the importance of ribosomes? Let me start with ribosome. Ribosome. What is the importance of ribosome? I'm starting with ribosome. What is the functions of ribosome? That is what I'm starting with. Excellent. It's used for protein synthesis. So you see, you have your mark here. So then, endoplasmic reticulum. What is the functions of endoplasmic reticulum? So we are done with the ribosome. So ribosome is used for protein synthesis. So all the proteins that are synthesized in a cell is due to the work of ribosome. So now, what is the functions of endoplasmic reticulum? The function of endoplasmic reticulum. No, I'm talking about the functions of endoplasmic reticulum. No, I can agree with lipid synthesis, but not protein synthesis. Lipids, lipids biosynthesis. So I can accept this. It is used in lipid biosynthesis. And I can also agree it's important in lipid biosynthesis and transports of materials around the cells. So I will add with this. Endoplasmic reticulum is used for the lipid biosynthesis transports and transport of materials around the cell. So see all these things, even if you don't know them before, please you should be knowing them now around the cell. So it is involved in the synthesis of protein and the synthesis of lipids and also transport of materials across the cell. So then now, what is the functions of lysosome? Lysosome. So we now mention the functions of ribosome, endoplasmic reticulum, and then the next thing is lysosome. So what is the functions of lysosome? The functions of lysosome. The functions of lysosome. Excellent, Mariam and Hassan. It helps in the destruction of cellular debris. You can say just removal is important in the removal of cellular debris and foreign organism like bacteria. So what I want you to understand is that the major functions of lysosome, it removes cellular debris, and also if there is any foreign substances like bacteria, viruses, or any organism that can cause disease. So these lysosomes is highly rich in an enzyme called lysozyme. Lysozyme. What is lysozyme? It's an enzyme that is found in the lysosome. And the major importance of these lysosome is to remove harmful substances, destroy them, and eliminate them. It can be a cellular debris, it can be an organism or foreign organism that can cause disease in our body. Yes, detoxification, you can also mention that. So you can removal of cellular debris and foreign organisms like bacteria that can cause disease. So you see, now we mentioned actually the functions of lysosome. When there is any cellular debris or any substances that can cause damage or that can cause harm to the cell. So this lysosomes is like a soldier that is found in a cell. It's like an army that found in a cell that trying to detoxify or remove any substances that can cause disease in a cell or that can affect the function of the cell. So that is the functions of lysosome. So you see we mentioned three. So then the next thing is nucleus. What is a nucleus? What is the functions of nucleus? The functions of nucleus. The functions of nucleus. Nucleus. Please, I want to hear from you so that I don't want to only be alone to be answering this. Yes, nucleus it control all the activities of a cell, all the activities of a cell. And you can also add that it contains genetic materials, e.g., DNA and RNA. So I think if you can mention this for the nucleus, I think you have done well. That nucleus, it control all the activities of the cell. It control by the nucleus, and very importantly, it contains genetic material like DNA and RNA. And you shouldn't forget, ladies and gentlemen, I told you that DNA is a language of life. So that is why, because DNA is responsible for coding any uh, organelles, one, the cellular differentiation and specialization is all controlled by DNA. So that is why DNA is considered as a language of life, and it's because it is in the presence of, it is found in the nucleus. That is why the nucleus control all the activities of a cell. So then the next one, which is the last one, question 5, the functions of mitochondria is considered as a powerhouse of a cell due to energy generation. Due to energy generation in the form of ATP. Due to energy generation in the form of ATP. So it is called powerhouse of a cell due to energy generation. Due to energy generation. So because it generates energy, so because of this energy generation in the form of ATP, that is why nucleus is considered as a powerhouse of the cell. And what is the full meaning of ATP? 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 Excellent. Adenosine triphosphate. So I think, ladies and gentlemen, we have answered question. Yeah. So let me share the question paper again. Able to answer question 1A and B. So you see question 1A carries, check very well, it carried the number of marks there for question 1. Question B is 4 marks. So each of these, if you are able to describe their functions, mark, and then the question 1C said two differences between nucleotides and nucleosides. The differences between two differences and nucleosides. So what are the two differences between nucleotide and nucleosides? So now, what are the differences between nucleotides and nucleosides? So who can tell me what is first? We start with the composition. What and what form nucleotide? What are the composition of nucleotides? The composition of nucleotides. What are the composition of nucleotides? What are the composition of nucleotides and nucleosides? Excellent. So the nucleotides is made up of nitrogenous bases plus pentose sugar plus phosphates. So that is the first differences in terms of composition. So the nucleotide is made up of three composition. We have nitrogenous bases, pentose sugar, and phosphate. But for nucleoside, it's made up of course, nitrogenous bases plus pentose sugar. So this is the first differences between nucleotides and nucleosides. Then another difference is nucleotides are building block of DNA. Nucleotides are building block of DNA, while nucleosides is a component component of nucleotide. So please now, do you understand the difference between the two? Nucleotide is made up of nitrogenous bases plus pentose sugar plus phosphate, Nucleoside is made up of nitrogenous bases and pentose sugar only, while nucleotides are building blocks of DNA, while nucleoside is a component of a nucleotides. So this is the difference between the two. So now we answer question 1 correctly. Let me actually look at question 2. Lisa, can you list more differences? No, actually there is no much differences between these two. So now question 2a says describe the plasma membrane of a cell. Describe the question says the differences between nucleotides. The nucleotide is made up of three components, which is nitrogenous bases, pentose sugar, and then the nucleoside is made up of only nitrogenous bases plus pentose sugar. The nucleotides are the building blocks of DNA, while nucleoside is a component of nucleotides. So now, let's look at question 2a. Describe the plasma membrane of a cell. Describe the plasma membrane of a cell. Question 2a says, describe the plasma membrane of a cell. So, how are we going to describe the plasma membrane of a cell? The plasma membrane of a cell. First, to describe the plasma membrane, we are now going to first, I think at this point, yeah, excellent, Mikael Umar. We can start with the function, but actually it's not the functions that we are looking for. So you can now see, describe the plasma membrane of a cell. So we can now say plasma membrane, it is a membrane that control the transport of material. So plasma membrane is a phospholipid bilayer. First, this is the most important thing that you need to know, because we are to describe. It means that we have to describe it structurally. So plasma membrane is a phospholipid bilayer that control the movement of materials that control the movement of materials in a cell. So that is the first description. So to describe the plasma membrane is a phospholipid bilayer that control the movement of materials in a cell. So you start with that. Excellent. So now we have to, I think to me, we have to come back and describe the phospholipid bilayer. So the phospholipid bilayer, it is a layer that comprises of two components. It comprises of two components. What are those components? Hydrophilic heads and hydrophobic tail. So what is the phospholipid bilayer? So first, ladies and gentlemen, we did you do remember that a phospholipid is an example of a complex lipids. It's an example of complex lipids. So how is this complex lipids are? Excellent. So this is a phospholipid, just a brief description of a phospholipid. You see, this is the alkyl group, this is also another alkyl group, and this is the phosphate group. So this is a phospholipid bilayer. So if you look at it from here, ladies and gentlemen, from here to here in this phospholipids, this is called hydrophobic. Hydrophobic tail. And then from here to here it is hydrophilic tail. Hydrophilic tail. So I think, ladies and gentlemen, what I want you to understand, it is very important. Go to your textbooks and I would watch from some of my videos. I actually, okay, someone is saying that the word is not clear. The problem, is it from me or from your side? Please, can you clearly see my box? Okay. So now what I'm saying, this is how the structure of phospholipid look like. It has two different other side chain. It has two other side chain. So we have R1, R2, and we have phosphate group attached here. So the part of the phosphate group is hydrophilic tail. Sorry, hydrophilic head. It's not tail, hydrophilic head. While the other part is hydrophilic tail. So in describing the phospholipid bilayer, so you can now say that first, plasma membrane, it is a phospholipid bilayer that controls the movement of materials in a cell. Then full stop. Then this phospholipid bilayer is made up of hydrophobic tail and hydrophilic head. Are you getting my point? First, describe the structure or describe the membrane of a cell. First, a membrane is a phospholipid bilayer that controls the movement of materials across the cell or in a cell. First. Then second, the phospholipid bilayer is made up of hydrophobic tail and hydrophilic head. I think you understand. So this hydrophobic tail is embedded in embedded into the cell, is embedded into the cell, while the hydrophilic head is exposed on the surface of the cell. So you see, you are describing first, it is a phospholipid bilayer which is made up of hydrophobic tail and hydrophilic head. Then the hydrophobic tail is embedded into the cell, while the hydrophilic head is exposed on the surface of the cell. So if you do this, you are describing. So this is how you can describe the cell membrane. Yes, it is a double bound. That is why it is called bilayer. It means it is. So now, the next thing after this description, what next? Then you can now see the composition of the cell membrane. So after you finish with this, then you now come with the composition. The composition of cell membrane, that is CM. So who can tell me what are the composition of cell membrane, please? What are the composition of cell membrane? Who can tell me what and what do we have in a cell membrane? The composition of cell membrane. Who can tell me? I'm waiting for your answer. No, 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 no. Excellent. Glycolipids, that is number one. We have glyco. Number one, we have glycolipids. Then two, we have glycoproteins. Agree? Then what again? Yes, I agree. Protein. We have proteins. Then what again? Four. Phospholipids. Then what again? Cholesterol. So I think even with these five, ladies and gentlemen, please you should know that what are the composition of cell membrane. So you started with this, that a cell membrane, it is a lipid bilayer that control the movement of substances across the cell membrane, and this lipid bilayer is made up of hydrophobic tail and hydrophilic head. Then the composition of the cell membrane, then you mentioned this. So now my question is, among these five major composition of the cell membrane, which one has the highest percentage? The one that is abundant on the cell membrane among all these five: glycoproteins, glycolipids, proteins, phospholipid, and cholesterol. It's not cholesterol. Please, actually, I'm shocked that up till now, many of you are yet to join my YouTube membership. Although I want to tell you that actually from today, every day we are going to solve past question paper. Every day we are going to solve past question paper. So tomorrow we'll also solve another past question paper. So if you are not a member, as I always said, you will not have access to my physiology video. You will not have access to physiology 225, physiology 228, biostatistic, and anatomy video. So yeah, Vivian, I know that you are a member, but it is expired, so you have to renew it. I think you have to renew it. It's very important, especially at this point. And what I want you to understand is that my YouTube channel comprises of courses from that we are going to do second semester. So we are going to start immediately after you write your exam. And then second, we are also going to continue with what do you sorry, five courses. I'm going to continue with five courses for your second semester. Biochem 242, human physiology 228, 226, anatomy 212, and microbiology. Yes, of course, Aisha to Ali, your own is you are still a member. So what I'm saying, ladies and gentlemen, if you are not a member, try as much as possible to be a member, because especially at this time, not actually because of, yeah, of course you can't download them. If you can download them, it means anybody can get the videos. So that is why it is unique, and it is only, no, no. Why would I have a lot of business and a lot of things. So we already fixed Saturday and Sunday for biochemistry Akande. So other days I will try to solve human physiology past question paper, especially this time around because I had that. So I will still continue with the solution to biostatistic human physiology 225, human physiology 227, and anatomy. So I will solve all those past question paper. I will solve them, but as I repeat myself, only those that are member to my YouTube channel can have access to them because it's my extra work. So I don't think, I was shocked sometime just if you 1,500 or 4,000 to access the videos, and till later on when we fail some of these courses, that is when so many of you start crying, will start feeling bad, and sometimes it's because of in our, yeah, it's because of your inaction. Yeah, video, okay, Aisha Ali, your videos are very helpful, sir. Yes, I know. It's because it is helpful, that is why many of you have joined, and you see the benefit, especially when you comes for your second semester. And I know that there is no any student that can committed him or herself to my videos that will have problem. I know. Okay, don't get the money. Don't get the money. We not get the money. Okay, no problem. But you spend the money on data. Just 1,500 is not, will not give you 2 gig data. So to pay that amount for YouTube membership, something that will definitely help your life and pass your courses, I think uh, it's like you are doing suicides. And sometimes I heard one of you said that she went to read the biochemistry materials and she realized that the courses there are very difficult, and she really enjoyed some. Yeah, I know. It's because the videos I enjoyed, that is why a lot of people have joined. And seriously, I used to surprise and shock. You will end up of spending more than 200,000 for your courses just to feel 1,500 on something, just to pay 500, sorry, 1,500 or 4,000 for a month to watch a complete videos, something that can help your life. Sometimes what actually surprised me is not always about the examination, even the knowledge you are going to gain. It's not something that you need to, or that that you have to use at this point. So very surprising, especially for those that can. Uh, yeah, Kuru, you are already a member, so it is not yet time to renew. crew Elizabeth, it can clearly show that you are still a member. So no, you see, the last time when the result was released, so many of you don't know what happened. There was a massive failure. There was a massive failure. Yeah, the differences between 1,000 and 400, 4,000. There are still some videos at the level of 4,000, and if you didn't pay them, if you didn't, if you are not at that 400 level, there are, 400,000 membership, so there are some videos that you might not still watch them again. Like here, of course, I'm very sure for now I have completely covered biochem. Sorry, human physiology 225. I have covered complete biostatistic. I have covered 80% of human physiology 227. And for anatomy, I can say I covered, let's say, 60%. So please, for God's sake, something that if you can be watching, why will you have a problem? Yeah, passing examinations, that is why it's 1 month. It's 1 month subscription. It's 1 month. After 1 month. So now, let me go back actually to the class. So the composition of the cell membrane. The composition of the cell membrane we have, but why I'm always telling you this, many of you will not understand what I'm telling you till when you start writing your examination. That is when you start uh, uh, that is when you will regret of not joining, because of course the videos are clear, the videos are simple. Some of the videos in just 15 minutes, in just 15-minute videos, a very complex process will be explained. That is why I said I'm going to be answering past question paper, so it is better you join. And joining doesn't mean that even if you join, there are still second semester, and second semester videos are still there. So immediately after this semester, we are also going to continue with the second semester. So it is better you join so that it will help you to cover some of the videos before the exam, and then immediately after the exams you can start with the second semester. So it is better you join or you renew it. So now what I'm saying, ladies and gentlemen, these are the composition of the cell membrane, and the one that has or that is most abundant in the cell membrane is phospholipids. So what I want you to understand, each of these component of cell membrane, it has its own function. But I think if you can describe this from here to here, you are going to get your 5 marks, complete 5 marks. You're going to get your complete 5 marks. So, Lisa, can we have the complete video for human? I think I have sent them. I have sent them in the group. Even if I didn't send them, I will try to compile them and send it. Even for this biochemistry, I will try to do that. So this is actually, but I want to, if you can watch, if you watch one of my videos, sorry, my videos on this cell membrane, there is a video on a cell membrane where I explain the functions of each of these, the function of each of these components. Okay, so now let's look at the next question. Okay, so now um, question 2b, using a named example, explain the movement of materials by exocytosis. Exocytosis. So now what is exocytosis? That is what we are going to start with first. What is exocytosis? Let me tell you, reading past question paper sometimes is not healthy. Your major problem sometimes you don't listen. You see, when we tell you that I know how complex this semester are, I know how complex this semester is. Instead for you to start reading on time, you will not do. You see, that is why some of you, as we are doing this, you are trying to be getting some of this information, and you are also contributing in solving the question because you have gone through some of this video, you have gone through the material. So now what I'm saying, ladies and gentlemen, and some of you will be thinking that in 2 days or in 3 days you will cover. No, no, I don't think so. But you still have the time. You can watch all the video. It will definitely help you. So now what I'm saying, yeah, it's not today that I'm answering by Chem 262. So another time. So what I'm saying, ladies and gentlemen, exocytosis with a named example. So now what is exocytosis? That is what we are going to start with. Exocytosis is the extrusion of substances from the cell. Yes, we have what we call, we have different types of transport. So we have bulk transport. So when we say bulk transport, it means the transport of larger molecules, the bigger molecules. So we have endocytosis. The types of bulk transport we have endocytosis, that is one, and then two, we have exocytosis. So exocytosis is the ability of a cells to expel materials by fusing a vesicles with a cell membrane, thus releasing the content outside the cell. Excellent and brilliant, Angela. So exocytosis is the movement of larger molecules or larger particles by fusing the molecules with a vesicles of the cell membrane, which release the content outside the cell. So what happens? Let's say this is a cell. This is a cell. And let's say this is the materials that want to be out, that will be expelled out of the cell. So what will happen? This cell will invaginate itself like this. It will invaginate itself to form a vesicles which releases the particles outside of the cell. So it usually is a process that involves the removal of larger particles or molecules outside the cell. And examples of this is like maybe degraded bacteria. Like bacteria is one of the examples of particles or materials that can be removed from the cells using exocytosis. Remember, I told you we have a lysosome. So when maybe a cell is infected with a bacteria, these lysosomes will degrade the bacteria. It will degrade the bacteria, and the bacteria is a larger particle, it's a bulk molecule. So the removal of these debris or cellular debris of bacteria outside the cell is a typical example of exocytosis. So exocytosis is a pancreatic gland producing insulin that acts as a glucose and convert. So that is actually exocytosis. So I want you to go and look at it very well. I did a video on that with so many examples. So then that is this. So I think let's look at another question, although I think time it is going. So I'm still going answering this question paper tomorrow. But let me try to also answer uh he said mention any two amino sugars. So please, who can give examples of amino sugars? Two examples of amino sugars. Two amino sugars. Two examples of amino sugars. What are amino sugars? Amino sugars are sugars in which one of their OH group is replaced with amino group. It's a sugars in which one of their OH group is replaced by amino group. So when you have one of the OH group of the sugars it replaced with amino group, so that sugar is called amino sugar. Like for example, if OH group in glucose is changed with amino, it's now called glucosamine. Glucosamine. So if you change one OH group with amino group in glucose, it is now called glucosamine. So glucosamine is one of the examples of amino sugar. Another examples of amino sugar we have galactosamine. We have fructosamine. So these are the examples of amino sugars. So the question says we are to give two examples. So now I mentioned three. Can we stop here or we should continue? Excellent. N-acetyl glucosamine is also another example. Sawa'u Aliyu Kera. So Amina Aisha Abdul Karim. So can we continue or we should stop here? We just answered two complete questions. Okay, so let's look at question 3. Question 3A says, how is the genetic information encoded in DNA? How, or briefly describe is the genetic material encoded in DNA? So how is the genetic information encoded in DNA? So let me try to answer this. So remember, what you are going to start is that DNA is a sequence of nucleotides that code for functional proteins. So in a DNA, we have a sequence of nucleotides, and this sequence of nucleotides are responsible for producing a functional proteins. So how these functional proteins are encoded in the DNA? The nucleotide sequence in the DNA are usually converted into mRNA, and the process of converting DNA to mRNA is called transcription. And then from transcription, mRNA is converted into protein, and the process is called translation. So the best thing to explain this, ladies and gentlemen, excellent, transcription and translation. So the genetic information is encoded in the DNA through the central dogma of molecular biology. So DNA to RNA, from RNA to protein, that's transcription, translation, and replication. So I think since so many of you are saying that we should continue tomorrow, I think there will be no problem. So we are going to continue tomorrow, and I will try to see maybe how we are going to summarize it and finish tomorrow. Then on Monday, maybe we'll start with biostatistics. Although I have solved a lot of question paper on biostatistics. So if you are not a member, uh, oh, I don't know Osundu. So we are going to stop here. So till tomorrow. Thank you very much. And please, for those of you that are not a member, please try to join the membership before Monday. And you can even join today so that at least you can visit all the questions. So thank you very much and see you next.